ان شاء الله التقدم الثاني طالب علاء عادل محمد شكران. ده طبعا ابن الاستاذ الدكتور عادل على الفوران الله يرحمه، كان استاذ معانا استاذ. وعلاء النهارده ان شاء الله تقدم ماجستير في فيسيولوجيا المتكامله تحت اشراف الاستاذ الدكتور عماد محمود زياد، الاستاذ الدكتور حماد محمد اسعد، والاستاذ الدكتور نادر محمد الطاهر من معهد الأوضاع. اتفضل علاء وربع ساعه بالظبط.
This is titled in Neurogenic and Therapeutic Potential of Adipose-Derived Mesenchymal stem cell and or Taurine in Alzheimer in aluminum chloride-induced Alzheimer disease derived model. The aim of work to evaluate the controversial aluminum chloride-induced Alzheimer's rat model, and then to investigate the possible neuroprotective effects of adipose-derived mesenchymal stem cell in support and in the presence of taurine amino acid, and finally to determine the efficacy of them as a therapeutic agents for Alzheimer's disease in general. Hereby we have the statistics about Alzheimer's disease and the prevalence worldwide. By 2050, it's expected to reach 131 million, while in 2015, it was reported that 47 million were reported to be dementia patients. The most common signs of Alzheimer's disease are memory deterioration and mood swings and social isolation in the advanced stage. Neurophysiologically, the main two hallmarks of Alzheimer's disease are extracellular beta amyloid deposition and intracellular neurofibrillary tangles formed by the tau protein, which is hyperphosphorylated from the endoskeleton of the axon. Then, about our model of aluminium, which was reported to be present in higher concentrations in 80 patients of familial Alzheimer's disease, and then it was reported to be anticholinergic activity as a choline toxin. Additionally, it was reported to enhance and to increase the deposition of amyloid beta. Our first therapeutic agent is adipose-derived mesenchymal stem cell, which was reported to pass the blood-brain barrier and form in the injured tissue of the brain in the transgenic Alzheimer's disease rat model, which was reported by Kim in 2019. Our second therapeutic agent is taurine, which is the key player of different physiological processes in the body. Generally, it participates in the bile acid conjugation, the prevention of oxidation-induced injury, and the neuroactivity include neuromodulation and neurotransmission. And finally, the calcium influx. Our experimental design included 3 steps: the adipose-derived mesenchymal stem cell obtained from the chemistry department after several successful trials of isolation and characterization. But unfortunately, our aim yield was 20 million cells, which was very challenging. Our next step was the modeling of Alzheimer's disease, and then the treatment with the two therapeutic agents, and finally the analysis was held to place. In detail, we have modeling or induction of Alzheimer's disease, like neuroinflammation, by the usage of aluminum chloride. The dose was 150 milligrams per kilogram for 40 days, and then we had a withdrawal phase which lasted for 20 days without it, through which rats haven't received any treatment. And finally, the treatment by the start of the therapeutic phase, through which rats were administered Adipose-Derived Mesenchymal stem cell by the dose 1 million per rat in the tail vein, in parallel to having taurine amino acid with the same dose of aluminum chloride according to the literature given by oral administration. Our first test was the behavioral test before the sacrifice of rat. Our behavioral main test was the teammates, through which we primarily evaluated the deterioration of the memory in the induced rats compared to the control. The process was ethically approved by the Institute of Animal Care and US Committee at Cairo University. Our analysis included apoptotic markers through an anti-apoptotic markers, sedative stress biomarkers, pathology, beta amyloid in both the brain and serum. Statistical analysis were held by ANOVA, SPSS. Firstly, we evaluated the body-brain changes and the ratio between them. Here we find through the 3 successive phases in both body weight and brain weight were reduced in aluminum chloride treated groups compared to the control. On the other side, they treated with any of the 2 therapeutic agents individually or together, elevated or enhanced or reduced the effect of aluminum chloride, and but therefore increase the body and the brain weight. While in the Mesenchymal index, which represents the ratio between both, we find that aluminum chloride reduced the ratio in aluminum groups compared to control and compared to the treated, which represents amelioration effect of both the therapeutic agents. Here we have the teammates results comparing the activity of the rats of different groups through 3 successive stages. Both are almost similar. This is the first trial and this is the second trial. This is a combined figure. Those are the aluminum chloride groups, and the others are the white ones are controlled, while those are the treatment. Here we have the beta amyloid concentrations in the brain and serum for all groups. There were a significant increase in aluminum chloride groups compared to control, and significant reduction in all groups except the taurine group in the brain, which had no significant reduction. Regarding the oxidative stress, we have measured 4 parameters. Firstly, the MDA concentrations represented in this figure reflects higher concentrations in aluminum chloride groups compared to the control. Otherwise, in the treated groups, which witnessed a significant decrease compared to both the aluminum chloride group and the control groups. GSH followed a similar behavior almost. On the other hand, we had catalase and superoxide dismutase, which unlikely had an elevated concentration in aluminum chloride groups compared to the control, which might be, according to the literature, it was explained by the increase of oxidative stress regarding or due to the aluminum chloride, which by the way increased the free radicals of oxygen species. The only exception here is the Taurine group, stem cell group, which had it's statistically significant. However, it seems to be non-biologically significant enough. It's not hard, but please, the mode of expression or the gene expression of pro-apoptotic and anti-apoptotic markers included P53 and BUX as pro-apoptotic markers, which were demonstrated to have higher levels in aluminum chloride group compared to the control, while the treated animals showed a significant decrease comparing to both negative control and aluminum-induced mode. On the other hand, the expression of BCL2 as anti-apoptotic markers had an opposite behavior, which confirms the previous reading. In aluminum groups, which represent the induction-induced, model, it had a significant lower concentration or expression of BCL2 compared to the control. On the other hand, the treated animals had a very significant high levels of BCL2 anti-apoptotic marker. To ensure that our injected stem cell had reached the brain and participated in the amelioration of the neuroinflammation induced by aluminum chloride, 1 million cells were which were fluorescent dark, followed by the fluorescent microscope, and we found that they have passed the blood-brain barrier, which shows that the literature review, which was reported by Kim in 2012. Here we have the fluorescent histopathological investigations shows the normal here we have the normal tissue compared to the aluminum chloride induced XP. We have here hemorrhage and neuronal flux deposition here, and this is very obvious, the difference between this and this, which presents a highly toxic effect of aluminum chloride. Here we have here we have supplementary sections support the neurotoxicity of aluminum chloride, almost with different higher resolution. Here we have the treated group groups. Here we have Adipose-Derived mesenchymal stem cells, which appears to enhance the blood vessels recovery compared to the previous. And this is of the Taurine group, which is almost recovered, but not that much. And finally, this is the group which had administrative both Adipose-Derived mesenchymal stem cells and Taurine, which almost has a better conditions comparing to Taurine, but not the stem cell group or group. In conclusion, the present study investigated the potential therapeutic efficiency of Adipose-Derived mesenchymal stem cell and or Taurine, which by the way was reported to enhance the proliferation of neuronal stem cells in the hippocampus. And here are our main parameters, excluding the beta amyloid, which I forgotten to consider in this list. And here our reference, acknowledgement, our dear professors and supervisors who trust me. This research was funded by the Academy of Scientific Research and Technology and was hosted by the Animal Health at the National Research Center, and I have three travel grants by the International Brain Research Organization, which was very generous from that. Finally, any questions? Thank you.
كتير وعايز اصلا كلهم حطي ان هو مش على تفكير كده بس رغم كده انا عارفه ان هو فاهم كتير جدا. انا عارفه. شكرا جدا علاء واي حاجه. اسئله حضراتكم. طبعا لازم اديك فرصه للكام سنه ايه؟ ما طبعاش الموضوع ده اهم جدا.
هو اكشولي ارجع للسؤال. Let's start our work under the supervision of Dr. Merbert. We have isolated stem cell and we created them from human liposuction. But there was very challenging to reach. Then we had to take it from the
افضل ان انت يبقى نفس السبيشيز، يعني هو ليه مش صح. ده كان احنا لسه بنتكلم في البرزنتيشن قبلها طلب الفيزيولوجي احنا كمان دلوقتي في الشغل كده
to be perfectly honest, this is our reference to do that. They have used human Adipose-Derived mesenchymal stem cell for 15
ماشي بس مش مش صح
يعني
فكان يعني البرزنتيشن ده كان بيطالب ان احنا اشتغلنا ناخد ستام سيلز من نفس البيشنت، لكن ناخد سبيشيز ثانيه خالص يعني
اتليست يعني لما تتكلمش تعمل بومينج وتعمل ريجيليشن للبيشنت they have an effect on growth factors and some interleukin.
انا ليه بس تعليق على ترتيب الصور ان كله يبقى مع بعض. يعني الكامسس مثلا يبقى
الكامسس مع بعض
كنترول انتريتد تريتد يبقى كله مع بعض. هو ده المفروض بس عشان وقت السيس ان شاء الله ان شاء الله. الخطوه الثالث